In 1912, miners digging for guano in a Nevada cave found something that stopped the operation entirely: mummies, dozens of them. Some of the artifacts were 4,000 years old. When scientists finally extracted DNA decades later, the results didn't match what anyone predicted. In 2011, a forensic genetics team ran mitochondrial DNA from desiccated human remains that had spent the better part of a century in museum storage. The samples came from Lovelock Cave, a dry volcanic cavity in the Nevada desert. And when the sequencing returned, the lead researcher stopped. The haplogroup was wrong. Not slightly off, not a degraded read, not a known variant with an unusual distribution. It was categorically absent from every sampled Native American population in the Great Basin. The remains had already been radiocarbon dated, over 1,500 years old, meaning whatever population left them predates the tribe. Archaeologists said owned this land by centuries. They hadn't found what was missing from the record. They had found what wasn't supposed to exist at all. The question that follows isn't academic. If a genetically distinct population lived in this cave, left thousands of artifacts, and then disappeared without passing their lineage to any known living group, where did they come from? And what does it mean that we nearly missed them entirely? Lovelock Cave sits in Humboldt County, about 90 miles northeast of Reno. It's a single oval chamber carved by an ancient shoreline, roughly 60 feet deep and 30 feet wide at its mouth. For most of the 20th century, it occupied a specific and settled place in North American archaeology, a site associated with the Lovelock culture, a prehistoric basin people generally considered ancestral to or contemporary with the Northern Paiute. That was the model. That was what the textbook said. The haplogroup extracted in 2011 doesn't fit that model. It has no match in the standard comparative reference panels for Great Basin populations, no match in documented Paiute lineages, and no match in the broader catalog of North American ancient DNA compiled across hundreds of samples. The haplogroup result is a measured fact. What population it belonged to is inference. What that implies for the peopling of the Americas is, at this point, speculation. But the measured fact doesn't change regardless of how the inference resolves. The model doesn't need minor adjustment to accommodate this result. It needs to be expanded in a direction it has not yet gone. The bones were real. The timeline wasn't. To understand why that haplogroup result creates pressure, it helps to know what the accepted model actually says, because the anomaly only lands if the orthodoxy has first been made visible. The dominant framework for the peopling of the Americas traces back to a Beringian land bridge crossing sometime between 13,000 and 20,000 years ago, with populations spreading south along coastal and interior corridors. The Clovis First model, which once held that a single founding wave produced all pre-Columbian populations, has been significantly revised over the past 2 decades. Multiple migration waves are now accepted. Pre-Clovis sites like Monteverde in Chile have pushed the arrival date back further still. But even in the revised, more complex version of this story, there is no framework for a genetically distinct population occupying the Great Basin and leaving no descendants, no overlap with surrounding groups, and no trace in any living or ancient comparative sample. That's not a gap. That's a contradiction. What happens to a model when the DNA says the model is incomplete? The people who might have answered that question were not scientists. They were guano miners. In 1912, a commercial operation broke into Lovelock Cave to extract bat dung for agricultural fertilizer. The material they were mining through had been accumulating for thousands of years, and it had preserved everything inside with extraordinary fidelity. Organic material, fiber, hair, bone. Before any trained archaeologist arrived, an unknown quantity of the site's contents had been moved, damaged, or destroyed. What we have from Lovelock is what survived that operation, which means we are already working with an incomplete record of an incomplete picture. The formal excavations came in 1912 and 1924, led by Llewellyn Loud for the University of California and Mark Harrington for the Museum of the American Indian. What they cataloged was remarkable. Several thousand artifacts, baskets, nets, textiles, ornaments, cordage worked with evident skill. And among them, thousands of Tule-reed duck decoys, some still bearing real feathers, with painted markings intact after more than 1,000 years. This was not a seasonal camp. The craftsmanship and density of material suggest a population that returned to this cave across generations, that knew it well, that considered it worth maintaining. The DNA study came nearly 90 years after those excavations, which raises an uncomfortable question. How many other sets of remains are sitting in museum storage right now, never subjected to modern genomic analysis, waiting for someone to ask the right question? The specific tests use mitochondrial DNA extracted from bone and hair samples. Mitochondrial DNA is passed maternally, unchanged through generations, and because it exists in high copy numbers within each cell, it survives in ancient tissue far better than nuclear DNA. The haplogroup identification was run against the standard comparative reference panels for Native American lineages, haplogroups A, B, C, D, and X, which together account for the vast majority of documented Indigenous American mitochondrial diversity. The anomalous result was not isolated to a single sample. It held across multiple specimens from the same assemblage. Contamination produces inconsistent signals. It does not replicate systematically across independent samples drawn from the same site and the same cultural horizon. The lab wasn't looking for a mystery. The mystery found the lab. What the DNA cannot tell us on its own is whether this represents a founding population predating the known basin groups, a later migration wave that arrived and ended without descendants, or something that does not map onto either category. Those interpretive layers remain genuinely open, but the haplogroup divergence itself, the raw measurement, is not open. It is documented. It sits in the record. And then there's the isotope data, which is where the story stops being something a single methodological objection can dissolve. Carbon and nitrogen isotope, analysis of bone collagen reconstructs dietary patterns, specifically the ratio of aquatic to terrestrial protein consumed during the years bone tissue was forming. The isotope signature from the Lovelock remains suggests a dietary profile inconsistent with the documented subsistence strategies of known Great Basin groups at the corresponding period. The protein ratios point toward heavier reliance on aquatic resources than surrounding basin populations of the same era typically show. The genetics are anomalous. The diet is anomalous. The material culture is unlike anything recovered from surrounding sites in the same period. 3 independent lines of evidence, 3 anomalies, all pointing the same direction. The convergence matters because each method can be challenged individually. Contamination affects DNA. Reference panels have gaps. Isotope readings can reflect local environmental variability. But when 3 separate methods, each with its own distinct error profile, each measuring a completely different physical property, converge on the same anomalous result for the same population, simultaneously, the probability that all 3 are wrong in the same direction becomes very hard to sustain. 1 anomaly is a flag. 3 is a pattern. Here is where the story gets complicated, not scientifically, but culturally, because Lovelock Cave has a mythology that ran far ahead of the evidence. And that mythology has done real damage to the legitimate scientific discussion for decades. Early 20th century accounts emphasized the red or auburn coloration of some recovered hair samples. Later popular retellings built this into a sustained narrative about a race of red-haired giants, a pre-Columbian people of unusual stature supposedly displaced by ancestors of the Paiute. The giant claim is a significant exaggeration of ambiguous skeletal data. The individuals recovered from Lovelock were within normal human height range. The hair color, which is physically documented and real, has multiple potential explanations, including post-depositional chemical changes to pigmentation in preserved organic specimens, a well-documented phenomenon in arid archaeological contexts. The mythology is fabrication layered over genuine observation, and it has made the real anomaly nearly impossible to discuss without the conversation collapsing into fringe territory. The most destabilizing version of this story is not the giant story. It never was. The real version is stranger and more consequential precisely because it doesn't require exaggeration to be troubling. Lovelock doesn't stand alone. Across the Americas, ancient DNA work over the past 2 decades has returned results that complicate the standard peopling model with enough regularity that the pattern demands explanation rather than case-by-case dismissal. The Spirit Cave mummy, recovered from Nevada in 1940 and radiocarbon dated to roughly 9,400 years before present, showed morphological features that led some researchers to question whether it fit the expected population profile for the region. When ancient DNA was eventually extracted, after years of legal dispute under the Native American Graves Protection and Repatriation Act, the results confirmed Native American ancestry while revealing population structure that did not map cleanly onto modern tribal affiliations. The Anzac I child from Montana, one of the oldest genetically analyzed individuals in the Americas at roughly 12,600 years before present, carried evidence of population divergence and isolation events, suggesting the early peopling was more fractured and discontinuous than a single spreading wave would predict. The same pattern has emerged from ancient DNA work in Europe and Asia, populations that occupied regions for millennia and then vanished from the genetic record without detectable contribution to modern inhabitants of the same land. This is not a peculiarity of American prehistory. It appears to be a feature of the deep human past, almost everywhere that ancient DNA has reached sufficient resolution to reveal it. The most instructive parallel for what the Lovelock evidence may eventually require is Kennewick Man. In '96, skeletal remains were found eroding from the bank of the Columbia River in Washington state. Early morphological analysis suggested features some researchers interpreted as inconsistent with typical Native American skeletal profiles, triggering a decade-long legal and scientific dispute involving federal agencies, tribal nations, and competing research teams working under different constraints. Access was restricted for years. The science was delayed not by lack of method, but by the collision of jurisdiction, law, and institutional reluctance. When comprehensive genomic analysis was finally completed, it confirmed Native American ancestry, but also revealed population structure and genetic affinities, adding genuine complexity to the peopling story. Complexity that would not have been visible without the analysis that had been delayed for nearly 2 decades. Every generation thinks it has finally mapped the past. Every generation is wrong about at least one major thing. The Kennewick case matters here not as a direct equivalence, but as a structural demonstration. The scientific community has a documented track record of navigating contested ancient DNA results, initial uncertainty, institutional friction, restricted access, years of methodological dispute, and arriving at a richer picture on the other side. The discomfort of the process is not evidence that the process is broken. It is evidence that the evidence is doing its job. The scholarly criticism of the Lovelock DNA study deserves direct engagement because it is specific and not baseless. Researchers have raised concerns about contamination risk, given the sample's age and storage history, about the limited size of the reference panels used for haplogroup comparison, and about the difficulty of distinguishing genuine population-level divergence from sequencing artifacts in degraded ancient DNA. These are legitimate methodological cautions. They address the confidence level of the haplogroup assignment. They do not address the isotope data. They do not address the distinctiveness of the material culture. They do not address the radiocarbon chronology. The anomaly has multiple independent legs, and the criticism, as articulated, only reaches one of them. The critics are doing their job. The question the evidence keeps forcing is whether their concerns, valid within their own scope, fully account for everything pointing in the same direction. Even if the haplogroup assignment is revised downward in confidence pending reanalysis with current sequencing methods, a reasonable next step, the isotope signature still describes a dietary profile that does not match the known population of this region in this period. That question has not been answered. The criticism left it standing. The broader framework consolidating in academic literature holds that the peopling of the Americas involved not a clean sequence of discrete waves, but a more chaotic process, populations arriving at different times, via different routes, establishing themselves in specific regions, and in some cases ending without genetic successors. The term used in population genetics is ghost populations, lineages visible only as anomalous signals in ancient DNA, detectable in the gap between what the model predicts and what the sample produces. This is the human dimension of what Lovelock represents. Not giants, not suppressed knowledge. The possibility that people lived in this desert, people with language, ceremony, and enough knowledge of their environment to craft convincing duck decoys from marsh reeds, and that the only legible trace they left is a haplogroup matching nothing in our reference panels, and several thousand artifacts in a Nevada museum. The land holds more history than we have names for. The fact that a genetic line ended does not make the lives of the people who carried it less real. It makes their near-total erasure from our reconstruction of the past more significant, not less. We were looking for confirmation of what we already believed, and we nearly missed them entirely. Most of the Lovelock material has not been subjected to modern whole genome sequencing. The 2011 study extracted mitochondrial DNA, which provides haplogroup data, but not the full resolution of nuclear genomic analysis, the kind that can map population structure, detect admixture events, and reveal ancestral relationships at a precision haplogroups alone cannot reach. Enrichment methods now targeting ancient human DNA specifically, deeper coverage protocols, and substantially improved authentication frameworks have transformed what is recoverable from material of this preservation quality. The Lovelock assemblage, under current methods, is a different scientific resource than it was a decade ago. The question of what a comprehensive reanalysis would show has not been put to the material. Partially unexcavated, the cave itself is dark, dry, sealed in parts, waiting, not because the questions have been answered, but because they haven't been asked of the right layers yet. Lower stratigraphic deposit of Lovelock Cave, below the level that produced every remain and artifact examined in every study conducted so far, contains a sealed layer documented during the original excavations and never fully opened. Its contents are unknown. It is estimated to predate the upper cultural horizon by several thousand years, though that estimate has not been tested against modern dating methods because the layer has not been excavated. Whatever is there has not been touched, sampled, or asked anything. Haplogroup. Anomaly, the isotope signature, the duck decoys, the century of deferred questions, all of it came from above that layer. If this is the kind of history that refuses to resolve neatly, that keeps generating harder questions the closer you look, there is more of it here. But the question the evidence keeps pointing toward, without answering, isn't who the Lovelock people were, it's what is underneath them.